Hello, everyone. Today we want to talk about water molecules, their structure, their solubility, and something that we call the hydrophobic effect. So I'm going to share my screen, and I'm going to be off in the corner because I want to be able to show you some of these principles using a model kit. So I'm going to start from the beginning. So why is it that every biochemistry textbook has a chapter on water? Well, that's because water is central to biochemistry. Water influences the shapes and functions of biomolecules, so we're going to be very interested in structure function studies going forward. Water is a reaction medium, so you can see from the graphic here that we have lots and lots of water in our biological system. Water is also involved in reactions as a reactant or a product. But often the true reactant or product is derived from water as either H plus or OH minus ions. So what that means is the reaction depends on pH, and that's also very important as well. So today's four topics are the structure of water, water as a solvent, the hydrophobic effect, and osmosis and diffusion. So first, the structure of water. So what's being shown here is a ball and stick configuration of water with the oxygen in the middle, covalently bonded to two hydrogens in which the electrons are unequally shared. And superimposed on top of that is the van der Waals radius. So that's the electron cloud. So when we see ball and stick models, we should not forget that they're really not that small, that there are the electron clouds associated with these atoms and that they cannot get too close to each other or the negatively charged electrons will repel. So water is polar due to both its molecular shape and the electronegativity difference between the hydrogen and the oxygen. So we can see that we have this sp3 tetrahedral geometry. But if we look just at the oxygen and the hydrogens as forming the shape, you see that we have this angular or this bent shape. And here we have the electrons being shared with the hydrogens to form the partially positively charged hydrogens, and we have the two lone pairs over here. So water forms hydrogen bonds, and the reason it can do this is because since the electrons are unequally shared between the oxygen, which is more electronegative, and the hydrogen, which is less electronegative, we have a partial positive charge on the hydrogen and a partial negative charge on the oxygen. And this shows the strongest possible hydrogen bond where you have 180 degrees where you have your oxygen, the hydrogen, and the oxygen over here as 180 degrees. And this line right here, the dashed line with the yellow, is meant to show the hydrogen bond. So that's the attraction between the partially positive hydrogen on one water molecule and the partially negative oxygen on its neighbor. So I have a model kit here, and the model kit shows water molecules. So we can see that we're using CPK coloring, where red is oxygen and white is hydrogen. And there are some magnets here that allow me to show how water can form hydrogen bonds. And here, the model kit won't actually let me do it wrong. It will show how the partially positive hydrogen is attracted to the partially negative oxygen. So I can form a water molecule with these extended hydrogen bonds. The other thing I can do with this water molecule kit is I can show how each water molecule forms four bonds. So if I look at this water molecule right here, I can show where I can have 1, 2, 3, 4. So if you look at the water molecule in the center, it's forming hydrogen bonds with 4 of its neighbors. And that's because it has 2 hydrogens that can serve as hydrogen bond donors, and then it has those 2 lone electron pairs that can be hydrogen bond acceptors for 2 other water molecules. So that's shown in this diagram, and I can show that in this model kit as well. So water has a higher melting point than substances of similar masses. So if you compare with ammonia and methane, methane doesn't form hydrogen bonds at all. Ammonia does, but only forms 3, and the hydrogen bonds are weaker because the electronegativity difference between hydrogen and nitrogen is not as great as it is between oxygen and hydrogen. So you can see that the melting point of water is way higher than these other substances. And that means that we need a lot of heat energy to break those hydrogen bonds to get solid water to be converted to liquid water. So ice is a striking example of the cumulative strength of those hydrogen bonds as illustrated by that high melting point that I just showed you. But the other thing is that this picture shows that then when the water molecules get together in the structure of ice, they form this very open structure. And what that means is that that structure is less dense than its liquid form. Ice is it's one of the substances that expands on freezing. You know that if you've tried to put something liquid in your freezer and maybe it's expanded or exploded if the container was too full. This is nice for biology because in the wintertime when ice will form on ponds, it forms on the surface and that protects the aquatic organisms below. It would be very bad if lakes froze from the bottom up. So when ice melts, the tetrahedral structure collapses. You don't have that open structure. You have a more dense structure now. So that means that the liquid has a greater density than the solid, and that is very strange. Most liquids, the solid form is more dense than the liquid. So if I had pure ethanol, and if I was able to make solid ethanol, and if I put the ethanol cubes, say, in a glass, they would sink to the bottom. We're used to ice cubes floating. That's our experience. Water is the liquid with which we have the most experience, but it's not typical. So we should keep that in mind. So the next thing that I want to talk about are the kinds of intermolecular forces that form. So we can look at covalent bonds. So these are common bonds in biochemistry, and this is the amount of heat energy it would take to break those bonds. But what we're interested in today are these non-covalent interactions between different molecules and their neighbors. So we can have ionic interactions and van der Waals forces. Some textbook authors will collectively refer to hydrogen bonds, dipole-dipole interactions, and London dispersion forces and call them all van der Waals forces. And here's some typical examples and the strength, the amount of heat energy it would take to break these interactions. You can see that the strength here is quite a lot less than it is for a covalent bond. So let's continue talking about these one at a time. So this is from our textbook. So again, I want to focus on the ionic interactions, hydrogen bonds, and van der Waals interactions. Those are non-covalent forces. So let's talk about ionic interactions first. So in my model kit, I have a sodium and a chloride ion, and this magnetic, this is pretty strong actually. It's pretty hard for me to pull these things apart. So if I had a lot of these, I could show you a sodium chloride lattice. And sodium chloride dissolves in water, but it does so only because you have to be able to break these, and then the water molecules will interact with those. So I'll show you that in a bit. So for right now, I just want you to see that the sodium ion, which is the smaller of the two, the sodium ion here is blue and the chloride is green. There's a very strong interaction between the two. And that's something that we learned in general chemistry. But in biochemistry, we're going to see that we have molecules that within the structures have ionic interactions. So here we see that we have functional groups that are positive and functional groups that are negative. And here we even have a chloride ion that that contributes. So these dots right here show similar kinds of ionic interactions in hemoglobin. And again, just like the sodium chloride I showed you, we have full positive and full negative charges that form these ionic interactions. This is not an interaction over here. This is a hydrogen bond. And again, you can see that there are no full positive and negative charges here. So back to our graphic right here, let's talk about hydrogen bonds. Okay, so the important features of a hydrogen bond is that you need a hydrogen bond donor and a hydrogen bond acceptor. And the way you spot a hydrogen bond donor is the hydrogen has to be bonded to something that's more electronegative than it is. And in biochemistry, you can pretty much rely on nitrogens and oxygens. So nitrogens and oxygens are more electronegative than hydrogen. If a hydrogen is bonded to a carbon, that can't be a hydrogen bond donor. And that's because hydrogen and carbon have about the same electronegativity. So that's our hydrogen bond donor. The hydrogen bond acceptor, we have oxygen or nitrogen. Again, they are more electronegative than carbon. So what you end up with is the oxygen or nitrogen is partially negative because they are bonded to the less electronegative carbon. So here's my donor. So my donor right here will be either oxygen or nitrogen. So here's my hydrogen right here. So the hydrogen is partially positively charged because it's covalently bonded to an oxygen or a nitrogen. The oxygen and the nitrogen are more electronegative than the hydrogen. So this D right here, donor, it cannot be a carbon. So look for oxygen, look for nitrogen, not carbon. Over here, this is my acceptor, and this acceptor again is either oxygen or nitrogen. It doesn't show this in the picture, but the oxygen or nitrogen over here, this would be bonded to a carbon. And again, the oxygen and the nitrogen have greater electronegativity than the carbon. So that renders this oxygen or nitrogen partially negative. So now these two molecules can be attracted to each other. The partially positive hydrogen over here on one molecule and the partially negative either oxygen or nitrogen on its neighbor. So if I look at this graphic right here, these are the donors over here on the left side. So you see that all of these have hydrogens and they're all bonded to the more electronegative oxygens or nitrogens. These are the acceptors. So here you see oxygens and nitrogens, and it doesn't show this in every single one, so you can write this in if you want to in your notes, but all of these are bonded to carbons. And again, so these are partially negative on this side because they're bonded to the less electronegative carbon. These are partially positive because these are all bonded to the more electronegative oxygens and nitrogens. So we have a partial positive charge here, partial negative charge here, and we have this favorable interaction that we refer to as a hydrogen bond. So let's go back to our graphic. Van der Waals interactions, we'll put those into three types: dipole-dipole, dipole-induced dipole, and London dispersion forces. We could note that hydrogen bonds are really just a very special type of dipole-dipole interactions. So these forces are very important in biological molecules, and there are three different ways that they can form. You can form between attractions among permanent dipoles. You can have molecules with a permanent dipole can induce a dipole in a neighboring polar molecule. Or you can have a collection of nonpolar molecules and instantaneous dipoles can arise between them. So again, what we're looking at is we're looking at differences in electronegativity. I'm going to stand up because my office light turns off if I'm not moving around enough. All right, I am back. So let's talk about these three different possibilities. This is a dipole-dipole interaction. I have two molecules right here, and they have a carbonyl group. The carbon is less electronegative than the oxygen. Here the artist has drawn electron density as red. So over here you see more red because the oxygen is more electronegative than the carbon. And here's an arrow right here to show that this is the electron distribution is in this direction where you have more negative charge here and less over here. And if the same thing happens here, then what you have between them, they didn't draw this, but this space right here is the dipole-dipole interaction. And it occurs between a partially negative oxygen and this partially positive carbon. So this dipole-dipole interaction occurs. And again, this occurs between two molecules, neighbors, when you have electrons are shared unequally, so a permanent dipole exists in that molecule. Here's the second type. This is dipole-induced dipole. So suppose I have a molecule with this carbonyl group, And here's the electron density right here. But its neighbor over here is the methyl group. And again, carbon and hydrogen have about the same electronegativity. So what happens when they get near each other is that this electron density over here can cause the electrons to temporarily kind of move over to this side. So this functional group right here with a permanent dipole induces a dipole in its neighbor. And the artist here drew the arrow to be a little bit skinnier to show that the electron distribution isn't as great here as it is in the induced dipole as it is in the permanent dipole. And the artist here also drew the space as being a lot bigger to show that the interaction that forms between a dipole and induced dipole is not as strong as it was between the dipole-dipole. And here's the third type, and this is called London dispersion forces. So we have two methyl groups, electrons shared fairly equally, but at any different instant, it's possible that you could have more of electron density over here, and then that would induce the electrons to kind of move over here, and then you'd have, in an instant, you might have a partial negative and a partial positive charge. And here again, the artist has drawn the arrows very skinny to show that the electron distribution is not very lopsided. And the space between the two of them is also large to indicate that the interaction between the two is fairly weak. So London dispersion forces are the kinds of forces you're going to find in hydrocarbons because carbon and hydrogens have fairly equal electronegativities. Okay, so let's move on to water as a solvent. So solubility depends on the ability of a solvent to interact with a solute more strongly than either the solute particles or the solvent particles interact with each other. In biochemistry, we're concerned with two types of substances that water can dissolve, polar and ionic. So this shows the dissolution of polar substances in water. So in my model kit, I have an ethanol molecule right here. And the ethanol molecule has an OH group. And you notice that when I take my water molecules, that the water molecules can form hydrogen bonds here with this alcohol functional group. And if I tried to get a water molecule to interact over here with the hydrocarbon portion of the ethanol, there isn't any interaction. There's no interaction there. So ethanol will dissolve in water because the water molecules can form favorable interactions with the OH group, and it sort of drags in the rest of this nonpolar portion of the molecule with it. And in this drawing right here, various solutes are shown in black, and the favorable interactions with water are shown. Water is shown in red, and the hydrogen bonds are shown in these red dots. So, so water can form favorable interactions with molecules that have permanent dipoles, and if they can form hydrogen bonds, then that's even better. So I just showed you the ethanol molecule, and again, ethanol interacts with water. Its hydroxyl group interacts favorably with water, but the hydrocarbon portion does not. So I will invite you to go to Top Hat and review this material. So water dissolves polar substances. Water also dissolves ionic substances as long as okay, so here's my sodium chloride. So in order to dissolve this, I have to be able to pull this apart, and that costs me energy. But I'm going to get some energy back because I can take water molecules and I can surround this ion with water molecules. I can form this nice little shell of water molecules right here. So that's my chloride ion, and I was able to form this nice shell of water molecules around it. So not all ionic substances are soluble. Sometimes that energy that it would cost you to break apart that ionic bond is too strong and you wouldn't get back enough energy when you formed these nice favorable interactions right here. But with sodium chloride it is. So sodium chloride is soluble. So notice both in the drawing and in this model kit that the chloride ion, which is green both in my model kit and on this picture, the chloride is negatively charged. And notice that it forms favorable interactions with the hydrogens of the water molecules because the hydrogens are partially positive. And the opposite is true with the sodium, sorry. If I have sodium, here's my sodium, and when I form the favorable interactions here, notice that with my sodium ion, the interactions are forming between the oxygens, which are partially negative, and the sodium is partially positive. So, I can get another one right here. So I can surround my positive ion with these water molecules. So I want to challenge you. I want to remind you that whenever you see ions in solution, they're not just Na pluses and Cl minuses. The Na plus looks like this, and the Cl minus looks like this. They're surrounded by shells of water molecules. And that's the same with ionic functional groups and biological molecules too. So whenever you see them, it won't be drawn this way, but remember you'll have this shell of water molecules surrounding them. Okay, let's talk about the hydrophobic effect. So the hydrophobic effect is a good explanation to describe the things I've just been talking about. So the hydrophobic effect refers to water's refusal to interact with nonpolar molecules. So these nonpolar molecules are called hydrophobic, water fearing, but it's really the other way around. It's water fears it, maybe. So nonpolar molecules interact with each other via those weak London dispersion forces that I just showed you, and typical examples are the hydrocarbons, but they do it not because they want to, not because these London dispersion forces are strong, because they're typically not, but because water refuses to interact with the nonpolar molecules, so they have no choice. So here's an example right here. So the nonpolar molecules right here are shown in gray, and if you have a nonpolar molecule, if you think about oil and water, so if you pour oil and water in a container, the oil will float on top of the water because the oil is less dense, that's why it floats. But the water and the oil don't interact with each other, and where they do interact is at the interface, and that's minimizing the surface area because water doesn't form favorable interactions with nonpolar molecules. And if you forced it to somehow, you would get these ordered cages around the nonpolar solutes, and that is not in accordance with the second law of thermodynamics. So the nonpolar groups aggregate, they minimize their contact with water, that minimizes the surface area, and it maximizes the entropy of the system, because the water molecules in the bulk solvent can form hydrogen bonds with each other, and they are in constant motion, so they are very disordered. So what is not favored is shown over here in part A. So here, hypothetically, we're hydrating nonpolar molecules. So the nonpolar molecule is shown in green, and the hydration right here, the hypothetical hydration, is shown in orange. So this would give less opportunity for the water molecules in the bulk solvent to form hydrogen bonds with themselves. And this structure right here is not favored, and it's also very ordered. So if you look at what's favored, here the nonpolar molecules are interacting with each other. You can see the interactions, you can see the orange right here minimizes the contact. So you have fewer interactions between the water molecules and the nonpolar molecules, and more hydrogen bonds can form among the water molecules and the bulk solvent. So this situation right here favors the second law of thermodynamics, whereas this is too ordered and it doesn't. So what I can do with my model kit is, as I showed you, I have this ethanol molecule right here. I can pop off this OH group, and the model kit has another hydrogen. So I can convert the ethanol to an ethane. And now, if I tried to form interactions between the water and the ethane, you can see I don't have favorable interactions at all. And in fact, one of the things you can do in the model kit that's kind of fun is you can take all the water molecules that are given to you in the kit, and you can put the ethane in it, and then you can take the cup and then kind of shake it up. And if you pour it off on your desk, you will find something that looks like this. You'll get the ethane by itself, and you'll get all of the water molecules forming hydrogen bonds with themselves. And that's what happens in the solvent. So I will invite you to go to Top Hat and look at some questions there to test your understanding. We're interested in this in biochemistry because there are a lot of molecules that have both that we call either amphipathic or amphiphilic, depending on your worldview. These are molecules that have both polar or nonpolar domains, and they're simultaneously hydrophilic and hydrophobic. So amphi means both. Pathic means to fear. Philic means to love. So both of these molecules are synonyms. And they describe molecules that have both polar and nonpolar domains. So amphi means I love both, or I fear both. But anyway, so I can give you some examples right here. So palmitate and oleate, these are soaps, and they have a polar portion, which is shown here in red, this carboxylate group, and then this hydrocarbon portion right here. So we can indicate this more simply by saying that these molecules have a polar head group and a nonpolar tail. And what happens if I put them in water is they form something called a micelle. And again, this blue right here is meant to represent the water. And water forms very favorable interactions with the polar head groups, but it doesn't with the nonpolar tails, as we just saw from the drawings and the model kits. So that means that the nonpolar tails have no choice but to interact with each other, again, weakly, via those lended dispersion forces in the center of the micelle. So here's another picture. So soaps and detergents are amphipathic, again, or amphiphilic substances. And the way that they clean is if I take some soap and put it in water and then kind of swish it around to form some suds, I form my micelles. And it turns out that grease and dirt are nonpolar molecules. So the dirt and grease will migrate to the center of these micelles. And since a lot of the soaps and detergents, these polar groups are often negatively charged, that means that you'll have a suspension of micelles that won't aggregate. They'll be suspended and they'll be away from each other because they'll be repelled by the negative charge. So you can easily wash away the micelles. So last, osmosis and diffusion. These are colligative properties. So remember from general chemistry, the colligative properties are properties that depend on the concentration of the solution, of the solute, sorry, and not its identity. And the examples we used in general chemistry are freezing point depression, boiling point elevation, osmosis, and diffusion. In biochemistry, osmosis and diffusion will be the ones that we are the most interested in. Osmotic pressure can be illustrated this way. If I take a solution of water and I dissolve either some salts maybe or a polar solute, and I stretch a membrane across that's permeable to water but not to the substance in here, and I have this my water levels are equal, and if I go away and I come back the next day, I'll see that the water has migrated into this tube, and you can see that the column has increased in height. And it does this because it's an attempt to equalize the concentration on both sides. So when we say osmotic pressure, imagine that the pressure I would have to exert to prevent this movement, that's called osmotic pressure. So we can illustrate it here with these examples of red blood cells. Red blood cells have a sodium ion concentration of 140 millimolar. if I put them in an isotonic solution, that's also 140 millimolar. And what that means is that I would have water moving in equal directions, both in and out of the cells. Suppose I would put my red blood cells in a hypertonic solution. That means the sodium ion concentration would be greater than 140 millimolar. And what that means is that water is going to move out. And again, it's an attempt to equalize the concentration to make this more concentrated and this more dilute. In a hypotonic medium, the cells would burst because, again, the water is going to move inside, and again, in an attempt to make the concentrations of each of these equal. And what happens here is that the cells will burst. We can use this same concept in dialysis. So here I have a solution. And say I have some protein that is represented by the purple molecules and some kind of smaller salts maybe that are represented by these red circles right here. If I put this in a buffer and stir it and leave overnight, what I'll find, as long as I've chosen my dialysis tubing carefully, the dialysis tubing has pores that allow the passage of the smaller molecules, but not the larger ones. So this is a way you can rid your solution of these red ions or small molecules if you want to. And you can do this 3 times, where I could take this my solution in my dialysis tubing, I can throw this solution away and put it in fresh medium, and then equilibrium will be established by the red ions moving across. And if you do that 3 times, you pretty much will get rid of all of the red ions. So that is it for now. So go ahead and read the next section in chapter 2, and I will see you next time.